I talk? I'm very loud. Can people hear me? All right, good. Well, thank you for inviting me here tonight. I really look forward to this. I think this is a very interesting venue to see how to describe these things and how to talk about these things in a more public forum. Often we concentrate so much on talking to other academics, we lose track of what we're really saying, because it's easy to start to sort of talk in code. As a bioethicist, what I'm always brought in to do, I'm always the last speaker, and I'm always the one that's supposed to throw the water on everything else and say, well, you really can't do that, that's not really right, and there are really all these problems, and that's exactly what I'll do for you today. But I have to say, Garret has set this up for me very nicely, because a lot of the issues that I wanted to talk about were some of the things that he touched on toward the end. One of the things that got me going when I was trying to do some reading about this topic was the incredible sense of optimism that people had, and the sense that personalized medicine was going to lay the foundation for the next wave of how healthcare would be organized in the United States. And Barack Obama, in fact, was the author of a bill that has been passed in the in the Senate, actually in Congress, before he was president, to initiate genomic and personalized medicine as a new paradigm for the healthcare system in the United States. The former Health and Human Services Director actually described the transformation toward personalized medicine he said it will improve the health of our nation in ways that were not imaginable less than 50 years ago. So when I started reading this, I thought, well, okay, what is it that personalized healthcare is supposed to do that's going to be so important and so different? And what was often mentioned in the bill and in various talks that people gave was that it's going to make it cheaper and it's going to make it safer. Sorry, I never get through talking. So what I want to look at very closely is, what do they mean by that? How is this approach going to make it cheaper and safer? And the approach meaning that they can do a genetic analysis of you, and on the basis of that genetic analysis, they're in a position to decide whether you should or shouldn't get a certain drug and how that drug should be dosed, or whether or not there's certain kinds of preventive measures that you would be recommended to take. So what has been established, I think, with the last talk is that the problem is that this is going to be very complicated. This is going to be very hard. And a lot of the way it's presented in the media is that it isn't that hard, that it's just flipping a switch, as Garret kept saying. And so how is it going to be complicated? This is a quote from Francis Collins, who's the former director of NHGRI. And what he says, he's looking specifically at pharmacogenetics, which would be the part of the process where they try to titrate or try to choose a drug specifically based on your genetics, as opposed to the preventive measures, which wouldn't be pharmacogenetics. But the sense in looking at the future of personalized medicine has long been that it is in pharmacogenetics where you're going to see the most progress the fastest, because it's more amenable to experimentation. And some of the earliest things like warfarin seem to be very promising in this way. So when they talk about saying that costs are going to come down, what they're saying often is that the cost to develop one of these drugs will be less expensive, okay? And the cost of drug development obviously is an enormous expense in healthcare, so that could matter. But the other issues that come up are, if these tests are so complicated, or let me rephrase that, if the differences among people are fairly subtle, and if it turns out, as research has shown, that you have to have a lot of these, you have to have a lot of differences to make a big enough difference that you can see it in a test, it's going to end up that creating the information for physicians to use, getting this test to the point that it could actually be used in a clinical setting, is going to cost an enormous amount of money, that there's going to have to be a lot of research and a lot of work done to create them in a format that people can actually use them. And what they have to ask in order for a test to be clinically applicable are questions like, does it identify people who aren't symptomatic? Does it find the obverse people? What happens with the outcomes in healthcare if you use this test? Are there risks that we don't see? And so there's a lot of research that is going to have to happen before any one of these tests can actually be used. And when people talk about costs going down because of the use of personalized medicine, they tend not to talk about those costs. The other thing that people tend not to talk about is the difficulty that the FDA is going to have in terms of regulating these tests. It's going to be very difficult to study them, say post-marketing studies once a drug is out in the public and the public is using it. How are they going to evaluate whether or not it's done what it's supposed to do? Because the question is whether or not something's been given to whom was supposed to get it. And in order to know if it was given to somebody who was supposed to get it, you're going to have to be able to look at their genotype to evaluate back how things were, how the drugs were handed out. And all of those things raise questions about the sort of broader administration, the practical challenges that exist with integrating personalized medicine. So it suggests that it is not going to be it's not going to be cost saving quite in the way that people have predicted. The other part of the prediction is that that there will be an improvement to safety. And what they're saying again there is very specifically, there are certain drugs that it's best to know that what a person's genotype is, specific genotype, because the drugs can hurt the person or they can just not be effective. And so what people are focusing on when they say that they can improve safety, it's that idea. It's the top idea there. But safety in the actual clinical implementation of a test has a broader definition that has to be engaged and has to be understood. And so, again, back to the complexity of these ideas and this research, the complexity of the processes, in fact, that personalized medicine is trying to access and use. One wonders how easy it'll be for physicians to actually know when a test is the right test to give to a patient. And there's been research done a while ago on how well physicians can use information about colon cancer testing. And that kind of test, the way that test was set up, was much more straightforward. And studies showed that over 30% of the physicians couldn't properly interpret the results of the tests that they had ordered. So that begs the question, which has been mentioned before in this work a lot. If the physicians can't really explain it, what's the likelihood that the patients are going to really understand their choices? And so what's the likelihood that informed consent of any kind is going to be adequate? And that begs the question sort of in a broader sense of what are the issues of safety. So again, in a narrow sense, you could imagine personalized medicine improving safety in some ways, but in other ways, it won't improve safety. It may, in fact, counteract some of the, it'll counterbalance some of the things that it'll be able to also improve. So again, if we move forward from this expanded notion of safety, one of the issues that has been most consistently discussed in terms of personalized medicine has been the question of confidentiality. So obviously, if a patient is going to be treated based on his or her genotype, they have to have genetic testing. And from the beginning, well, let's say from the beginning of breast cancer genetic testing, which was commercially available in about 1995, one of the major issues that patients coming in deciding whether or not to test, one of the major issues, one of the major reasons that they say, no, I don't think I'm going to do that, is they're concerned about confidentiality. They're concerned that their genetic information will somehow circulate, will somehow be revealed, and that on the basis of that information, they'll be perhaps discriminated against in a job they might be discriminated against in terms of getting information, getting insurance. And so for years, there has been this very serious concern, and so urgent action, a rogue scientist needs to be bypassed. Otherwise, the enormous research and clinical progress being made in the nascent area of personalized medicine will come to a crashing halt. So there's this sense that because people have been worried about confidentiality for so long, that if we're really going to make this shift to personalized medicine, which is based on genetic information, somehow we really do have to deal with this perceived public concern about genetic information. So Francis Collins used to be the head of the Human Genome Research Institute, made this one of his personal goals to have the legislation passed in the United States. And he finally did succeed, and he then retired, or temporarily retired at least. And last summer, the Genetic Information Non-Discrimination Act of 2008, also called Gina, but not Kolata, was passed. And there was great celebration among some people. And basically, it's a federal law that protects Americans from being treated unfairly because of differences in their DNA that may affect their health. I'm going to talk in a second about why this is controversial, but there's a more fundamental issue here, which is that as much as patients, research subjects, would talk about their concerns about confidentiality of genetic information and would cite that as a reason that they did not want to go through with testing. It's also the case that social scientists and lawyers and people in sort of independent think tanks have put an enormous amount of effort into monitoring the use and misuse of genetic information. And although the fact that they put the effort in doesn't mean that they necessarily have caught every instance where this might have happened, it does suggest that if there had been pervasive misuse of genetic information, that they would have caught it. And in fact, there's very little in research that demonstrates that there is misuse of genetic information. Doesn't mean it hasn't happened, doesn't mean it couldn't happen, but it's an interesting phenomenon that the way public concern or the way ethical and social concerns about genetic research and genetic medicine have been expressed is through this concern about confidentiality, when in fact there's very little evidence that that there have been problems with confidentiality. And one thing that's important about that is that it begs the question that if now you have this legislation, it begs the question as to whether it's actually going to make a difference. So maybe people will come up with yet another reason that they don't want to do genetic testing. So Gina is, in some circles, highly celebrated, and well, I'll show you some of the highlights. Let's just go into what it does. Okay, so it prohibits groups and individual health insurers from collecting and using a person's genetic information to determine eligibility and premiums. It limits employers' ability to collect genetic information, and it prohibits employers from using such information in making employment decisions such as hiring, firing, job assignments, or any other terms of employment. Now, in some ways, this is a rather impressive piece of legislation for the U.S. government. It's certainly very different from the medical record privacy legislation that passed a while ago, HIPAA, which was really very technical about how you could share certain kinds of information, but it didn't have really these sorts of protections in it. When it was passed, people wrote songs about it. Gina, you're supposed to sing this to YMCA. And there was a big party at which Francis Collins performed the song, Gina, for everybody. And so in light of that, one might wonder sort of where are the critics coming from? Why do the critics, are they not so happy about all of this? And there are a couple of issues. One, what I mentioned before, is that it's not entirely clear to people that passing Gina is actually going to address whatever concerns people might have. The other piece of it is, if you go back, it prohibits insurers from using the information from a genetic test to change somebody's insurance rates, to deny them insurance. They can't use it for job decisions. So if somebody has a breast cancer test for genetic breast cancer, and comes out positive, the insurance company can't increase her rate. Her employer can't fire her. But it does nothing if that woman 5 years down the line gets breast cancer. So at that point, it's state law that will decide whether or not the insurance company can raise her rates, whether or not the insurance company could, at a yearly cycle, cut off her insurance. And so the criticism of it is that it's not the sense that somehow Gina should have done it all. They should have, utterly reformed American healthcare. It's more the sense that it misses the point, which is that there are serious problems with the healthcare system in the United States generally, in its financing and the way insurance is set up. And so if the point is that the concern, the more general concern, the broader concern, is what happens to people who, when they get sick, can't pay for their health care. And so instead of having the question being, how can you make health care cheaper? How can you make health care safer, to which personalized medicine would be the answer? Instead, the question could be, how can we fashion a healthcare system that protects and promotes human well-being and does so not only for the select few, but for an entire population? Thank you.
Thank you, Pamela, for a real reality check. We're going to have a panel discussion now, and as the panelists come up to the podium, to their chairs, I will introduce the two other people that you haven't heard from yet. Dr. Celeste Simon is the scientific director of the Abramson Family Cancer Research Institute, and she's a professor in the Department of Cell and Developmental Biology at the University of Pennsylvania. She's interested in understanding the molecular basis of how a lack of oxygen can regulate the development of heart cells and stem cells, and how stem cells function, and also how oxygen availability can regulate tumor cell biology. And the other person is Dr. Sarah Tishkoff, and Dr. Tishkoff is a professor of genetics and biology at the University of Pennsylvania, and she studies patterns of genetic diversity in African populations, human evolutionary history, African and African American population history, and the genetic basis of disease susceptibility. So I want to welcome our other two panelists, and I guess they're going to pass questions up to me if you guys want to write them down on index cards. I have a few questions already, and I can start our discussion going with some of what I already have. One of the questions involves vaccinations. They say this person says that they believe that vaccines cause frequent harm to children. Is it possible that science will make it possible for every child to be genetically screened prior to vaccination? Is this a reasonable expectation in the near future, or have we over-promoted these? Signed, a concerned parent. I'm not sure who wants to take this, but it's an interesting question, I think. Okay.
At the risk of jumping into an area I know very little about, there is a perception that vaccines frequently cause harm to children, but that perception is not borne out in reality. There was an interesting study published by a CHOP scientist, Paul Offit, who helped develop the Bioethics vaccine, which really causes tremendous benefit to large populations of children. And he went through the various arguments and presented the best evidence that we have to date that to a large extent says that while there may be rare cases of reaction to a vaccine, the numbers are very, very small, and those are counterbalanced with the very large population that benefits from the use of vaccine. Is it possible to find genetic tests that would identify the children that would have a reaction to the vaccine from the ones that do not? That's a trickier undertaking in that we don't quite understand what would cause those reactions at present, but perhaps down the line there might be a way to combine genetic testing with susceptibility analysis.
Thank you. Anybody else want to chime in? Okay. I have another question. It says for Dr. Curran and or other panelists, I am discouraged by the apparent lack of progress in the prevention and treatment of sickle cell anemia since the molecular defect was identified more than 50 years ago. So is this going to tell us that it's harder than we think to try and make some progress here?
It's a very good question. Yes, we understood the defect in sickle cell disease a long time ago. We understood some of the gene mutations, and the gene hemoglobin attracted a lot of attention from many people who are very interested in using gene therapy approaches. It turns out to be way, way harder than anyone imagined. Not from a lack of trying. I know many scientists that worked extremely hard at trying to understand the regulation of hemoglobin so they could find a way to restore the corrected hemoglobin gene. Biology is very complex. It did come up in some of the other talks. It's not so straightforward. Hopefully, we are making progress, and some of the approaches that are being tested right now, including the one that I mentioned, will show better promise in the clinic. But it's a tough disease.
Yes.
I wanted to actually follow up on that because I just recently was made aware of a paper that came out in the journal Science, I think it was in December, in which they had, using some of these methods that you heard about, such as Genomewide association studies, they identified individuals who have very high levels of the type of hemoglobin that's typically expressed in the fetus, so-called fetal hemoglobin. And there seems to be some sort of genetic regulation that typically that gets shut down, and then the adult hemoglobin is expressed. And what they found is that people vary. Some people keep this turned on. And they actually found the genetic variants that seem to regulate that trait. And it seems to be a control region very close to the hemoglobin gene. And so potentially what that means is that they could come up with a mechanism to basically keep that gene switched on in individuals who have sickle cell. So they have an abnormal adult hemoglobin. They may have a mechanism. And other than that, I don't know how likely it is that they'll have clinical, applications for this. But I heard there was a lot of excitement about that.
Yeah.
If I could also say, I believe also in the same journal, Science, there are great mouse models of this disease, and one of the stem cell labs has been using blood cells derived from murine stem cells in a treatment methodology to cure a murine version of sickle cell as a proof of principle for how stem cell-based therapies might be used rather than gene therapy. This might be an application of taking totipotent embryonic stem cells, turning them into blood cells, which have been manipulated to correct that problem, and at least in an animal model it worked quite well. So, I think sickle cell is a fascinating example of knowing a lot about the disease but being unable to really treat it. But because things were implemented to try to approach that disease, it led to a lot of other diseases being treated better, and we've all benefited from that. But I think things have gone full circle now. Some of the most modern approaches are being brought to bear back on one of the best characterized molecular disorders. And so this is some of the uncertainty that we face in trying to treat these disorders. So just thought I'd mention that as well.
Yeah, it's interesting because the next question is about cancer genetics, and it's sort of, in a funny way, it kind of almost relates to sickle cell thing. My limited understanding of cancer genetics has failed to find specific changes in DNA for each cancer, so it's just the opposite. Instead of having one specific change, you know nothing to do about it. You have the cancers with, you don't have specific changes in the DNA for each cancer, this person's pointing out. BRCA1 and 2, those are the breast cancer genes, are famous genes, but only occur in a limited number of breast cancer patients. What is the risk of rolling out personalized medicine before we know causes? False negatives? Question mark? Another great question for the audience.
If you want, let me jump in there. So the variance that you see in the human genome is magnified in cancer. And although maybe some individuals predicted early on that we could identify the major subsets of cancer by a few mutations, it actually hasn't turned out to be the case. There are a large number of mutations. There are some pretty significant projects going on right now to try to map all of those genetic mutations in different types of cancer. Whole genome sequencing, for example, is being applied to diseases like brain cancer. And we're uncovering a higher level of complexity than we'd even imagined. So the best way to think about cancer is multiple diseases. Even something called breast cancer. There's not a breast cancer. There are many, many kinds of breast cancer. Sometimes a specific mutation will have a very dominant effect in certain cancers, and there may be a treatment that will be specific for that kind of mutation, but that will only apply well to the individual that carries that very particular mutation. And so really the issue for cancer is the diversity of genetics of all of those tumors, and we're trying to use genomic approaches now to sort through that complexity.
Is there less, I hate to take my prerogative here as a moderator, but is there less diversity in pediatric tumors?
People have said that, but we haven't studied it, so we don't know yet. The problem with studying pediatric disease is we tend to get the leftover funding after the big studies have been funded. Sorry, that's my
leftover drugs too, I noticed when you gave the talk.
The argument was made that since those tumors are rising early on, they may have not accumulated as much genetic diversity. But I know there are certain forms of cancer that we are studying in the lab, choroidal carcinoma, that have completely messed up the genomes with very great diversity. So I think until we study the individual diseases specifically, we probably can't address that question.
I could follow up though. Things are emerging from this systematic characterization where drugs were being used to treat, for example, lung cancer with, in a broad population, less than 20% efficacy, but then by carefully pinpointing not just which genes had mutations, but precisely what mutations they had, they were able to select patients that showed more like 80 to 90% response rates. So these kinds of things in tremendous detail, I think, will show benefit. But as Tom said, it's very complicated. And I think about the select because when they first discussed sequencing the entire human genome all those years ago, I just thought it was going to be extraordinarily expensive, and would it really, sequencing every single DNA base pair, would that really be beneficial? It's been hugely beneficial, and the same thing has been done for the human cancer genome atlas. And again, even though some people were so skeptical, surprising information has come out about how frequent some of these mutations turn up in a broader spectrum of cancers, and this can only help at least identify patients that are the most likely to respond to certain kinds of treatments, and I think that's only going to improve.
I think to address the part of the question that was about these personalized whole genome scans that you can get from 23andMe or DecodeMe, and is this going to lead to false negatives? I think it's a very important question because what you're seeing is the impact of direct-to-consumer testing. So in the lab, with scientists who are very sophisticated about these things, they understand the contingency of a particular finding and how many years it's going to be before they really know, and what the potential of knowing, which is very exciting, what the potential of knowing is, but they don't know now, and yet these companies still will give you a readout. I have a picture of one that's actually very interesting. And what's very important in terms of the false negative piece is the first time I saw this readout, it compared, one person went around and had themselves tested at Navigenics and DecodeMe and 23andMe. And each company actually uses different SNPs. They look at different parts of the genome for what they then present as the same condition. So if it's something about vulnerability to heart disease or something about vulnerability to osteoporosis or something, the disease or the condition is the same, but the SNP that they report on is different. So first off, it's just very confusing to a consumer what it means. But secondly, what's interesting is when there's an absence, when it says there's nothing found, how does the consumer interpret that? Does the consumer think, oh, well, I'm not vulnerable to heart disease, I'm not vulnerable to cancer? And already that was seen in breast cancer genetic testing, so that if a woman came back with a negative result, still means she has the same risk as any other woman in the population. But many women interpreted it to mean that they had no risk because they didn't have the gene. And so the sort of the science is on one track, and the public perception and the public presentation of the science is on a very different track. And I think it's a problem with direct-to-consumer testing, and it's a very good question of what is going to happen. And these tests are, at this point, not regulated because actually they claim that they're not medical, so they're not regulated under medical testing.
How hard is it for people to understand the probability estimate if someone says your probability of getting this disease is such and such? I wouldn't even know what that would mean and how worried should I be?
Yes, well, they don't actually tell you that when you get the test.
What do they tell you? Don't they give you like a probability?
Two of the companies just report whether or not you have the SNP that they chose to look at for a particular condition. So it's yes, no, you have it.
So you interpret that as meaning what? I have a 10% chance because I have this SNP, or I have a 100% chance.
They don't put a probability rating on it because they don't know. I mean, I don't on what basis. But
I don't know. I'm just wondering what it would mean.
And then another one actually has a genetic counselor that they give information to. I don't know how they interpret it.
Just in case you're wondering, I don't think anyone here has had those tests done, and I don't know any other scientist who hasn't done, apart from the people who like to see the genomes published on the web.
Just to follow up on that, brought up a good point, and that is to keep in mind that these places that do genetic testing like 23andMe and so on, some of the variants are very well established in terms of what the effect is. And some of them, they talked about, what was it, we were talking recreational genomics. Let's just find out for fun if we have the trait for, I don't know, something to do with the type of ear that you have, or if you could taste certain substances and things like that. And for some of these traits, it's very well known, there's a single known genetic variant. You can test it. It will be very predictive for whether or not you have that trait. But for many of the things that many of us in this room care about, which are the common diseases, hypertension, most types of cancer, diabetes, and things like that, at this point, we really don't know the genetic variants that predict with high success whether or not you're going to get those diseases. So people have done these studies, and they find variants associated with the disease in cases maybe just a little bit more than in controls. And so if the likelihood is equal, they say there's an odds ratio of 1. And for most of these diseases, I think the odds ratio is what, like 1.2 for cases versus controls. I mean, it's a very, very little risk. And yet what concerns me is that these companies are doing these tests, and the implication may be that, oh, you have this, it was associated in some people, now I'm at risk. And that's not at all the case.
I can tell you if you're at risk, then what are you supposed to do? Since if you're at risk for brain cancer, what am I supposed to do with that risk? You know, just wait? I mean, I don't know an early diagnosis for it. So it's, you know, some of these things are pretty tough. Maybe that's why nobody you knows as a scientist has had this done. What to do with the information. Here's a question for Dr. FitzGerald. Given the Bioethics experience voluntarily withdrawn by Merck, how can Celebrex remain on the market?
A lawyer in the audience. Well, I think I guess I should say, first of all, from my point of view, I think the issue is not to withdraw Bioethics or withdraw Celebrex. The issue is actually to do the science that will eventually lead you to be able to conserve the value of these drugs and manage the risk of these drugs. And unfortunately, there has been a lot less enthusiasm for investing in performing the science to get to that point, particularly after drugs have been approved and are making billions of dollars, which was the case with these drugs. So we need to get away from this sort of binary approach to both drug approval and drug withdrawal, where one day you have a drug that's not approved and suddenly it's approved and can be used by doctors for anything for unapproved uses. It's perfectly legal in any population, so at the discretion of the physician. Or tomorrow something bad happens and suddenly the drug is withdrawn, even though for 98% of the people exposed to it, they didn't have or weren't at risk of the problem that may have led to its withdrawal. So I think over time, as we indulge in the process of gradually personalizing medicine, we'll also indulge in the process of refining the way that we approve and withdraw drugs. So I think what happened with Vioxx and Celebrex is sort of interesting. Vioxx had a problem. You may ascribe motives that I'm in no position to do, but certainly a responsible thing in recognition of the problem was the withdrawal of the drug by the company, which in that case was Merck. Now, there may have been other reasons foreseeing legal liability, etc., but certainly the drug was withdrawn by the sponsor. And the FDA then considered at a meeting, did the other drugs in this class, like Bextra and Celebrex, cause a problem? And unanimously, the panel of experts voted that all three drugs caused a problem. They worked the same way. They probably caused the problem the same way. And then they decided what to do about it. And they voted about whether there should be a withdrawal or a reintroduction of Vioxx, for example. And the votes were divided. So I assumed that the next step in the process was the FDA listened to this, and then they went away and decided what to do. But actually the next step in the process is that the FDA enters into a negotiation with the sponsor, and the sponsor that was left at this point was Pfizer, which had Bextra and Celebrex. And I think what you got was the outcome of the negotiation. And the FDA was under a lot of fire, so they needed a scalp. The scalp was called Bextra. And the mitigating the damage to Celebrex, which remained in the market, was done by both leaving it on the market and then by applying the same black box warning to Celebrex as to all non-steroidal drugs, even though the strength of the evidence was asymmetrically implicating Celebrex at that point in time. So I think, as in so many other aspects of life, you had the outcome of a political process as opposed to something that was a sort of binary, scientifically driven decision. As in the personalization of medicine, social, political, economic, and scientific and medical factors came into play.
Another question sort of related, well, it's a pharmaceutical question, not it's related. What degree of responsibility does the pharmaceutical industry have in identifying the genetically responsive subpopulation prior to earning FDA approval for INDs? I guess that's for you, Dr. FitzGerald.
So I think, again, from a regulatory point of view, and I guess that was alluded to, we're at a stage of evolution here. And I think the real message you're hearing is enormous potential, some walking examples of the benefit to be had from this approach. But this is a process. It's not a peace treaty, it's not a declaration of war, it's a process. And that applies to the regulatory standards as well. So we're beginning to get some tests approved, genetic tests that can be combined with drugs that indicate a likelihood for, say, having an adverse effect or responding in an extreme way to the drug. But what we haven't got yet, even with those tests that have been approved, has been shown that they affect the outcome of practice. So I talked about warfarin, there are tests approved for warfarin, but people argue about whether these tests have actually added any value to just doing the sort of blood measurements that people have done for decades with warfarin to say you're having too much or you're having too little. And the real acid test will be, is there an added value that justifies the cost to the patient or the cost to the state as we move to a more comprehensive healthcare. So I think the regulation is in evolution as well. And you've heard how in recreational genomics, the regulation has by no means caught up with what's being pushed at you as consumers. I mean, I absolutely agree with Tom. I think if you're introspective, wealthy, or careless with your money, it can be entertaining to have these tests done. But the actual practical value, or how to interpret the information, is certainly beyond me. So I think, this is an industry that's crying out to be regulated at the moment, frankly. This personal genetic testing industry, and I think the regulation has to catch up with the process as the personalization events involve.
Can I just do a little follow-up? So suppose, for example, when we heard that something like Herceptin, the breast cancer drug, turns out to be effective only if you have a genetic test that indicates you're going to be susceptible. But when it was first tested, it didn't look like it was working because you had a mixed population, and they refined it and they found out who it worked on. Suppose when you had first tested Herceptin, you found a small effect, but that small effect really meant that some people got a gigantic effect and most people got nothing. Is it the responsibility of the drug company to develop that genetic test before they try to put this drug on the market?
Well, I think responsibility is a difficult thing to describe, except to other people. I'm no fan of Donald Rumsfeld, but those unknown unknowns will get you in the end. And, clearly from an economic point of view, it was cost effective for the drug company to do precisely those tests. So, given their vested interest in an economic benefit, never mind the benefits of public health, it's not for the want of motivation that they should fail to do that. But you've also got to be inspired enough to do the basic science that drives you to ask the appropriate question, that then leads you to perform the test, that then leads you to gain the evidence that allows you to meet the regulatory requirements. So I don't think it's so much responsibility as the inspiration and persistence to actually realize the benefit of this approach.
Dr. Sankar, assuming the individual is healthy according to genetic tests, does Gina prevent insurance companies from lowering health insurance costs for that individual and thus overall? That is, in what ways can we design a healthcare system that utilizes our combined genetic information? Interesting question.
I don't think it would stop them from lowering your costs, but wouldn't come up because the point is they don't have access to it at all. So they wouldn't know one way or the other whether or not you have the gene or you don't have the gene. But the point would be more if we return to population-based healthcare or insurance that we had 30 years ago where risk was pooled. It wouldn't matter whether it was genetic information or any other information. It would be that the risk would be spread across all people. And in that case, if you have a genetic problem that maybe evolves into a certain kind of, condition, you would be covered for that. If you had an accident, you would be covered for that. And the people who are healthy, they're very lucky that they're healthy. So I think that the way to get at what you're saying is to move into a different kind of financing that would allow us to not penalize people who have health issues, because in the end, we all have health issues of some kind.
But they would do things, just to be the devil's advocate again, they would do things like, you might say, let's say it's a life insurer, right?
Well, life insurance is very different from health insurance.
Oh, okay. So I'm trying to get my individual health insurance. I don't know how this works because I work for a company, but it's all pooled. But I assume do blood pressure, cholesterol, whatever. And then I come forward and say, hey, and guess what? I had all these genetic tests and I don't have any of the bad genes. Can I get a really great rate? Are they prohibited from, if I say, here's my stuff, here's my genetic information, from giving me a great rate because of that?
Yes.
They are
They cannot use your genetic test results to determine your rate.
Well, that's too bad.
You're very optimistic, aren't you, Gina?
I'm not having those tests. I don't want to know. All right, here's a really great question. How will personalized medicine play out in psychiatric diseases in which the diagnosis itself is tricky? For example, many patients have multiple diagnoses, bipolar, schizophrenia. There also are subtypes of diseases. Anybody want to take that on?
So it's still early days in the field of trying to identify the underlying genetic and molecular causes of many of those diseases. There is a belief that eventually we will identify some genes that predispose and may even give us clues to the underlying problem, which hopefully would open up new areas of therapy. But the whole field is fraught with the difficulty of diagnosis, because to do a genetic association study, you really need to know what you're comparing to what. Now, there is one terrible psychiatric disease in which we have a test, but it's kind of the worst case scenario. It's a disease called Huntington's chorea, which is associated with a very specific kind of gene mutation. It's an expansion of repeated sequences in the genome. Knowing how big that expansion is, you can almost predict the time frame in which someone will come down with symptoms. The symptoms generally arise in 35 to 40-year-old individuals, but they can arise much earlier and they can arise later. And there's a terrible degenerative cycle. There's no treatment, there's no cure, but the test is available. So why have it? Why have that test? I think it's a problem for families that actually have the gene within the family.
I was going to say that for many of these complex diseases, like psychiatric disorders, they're almost certainly not going to be due to changes in a single gene. And just as a case in point, there's been a lot of research being done in autism, and I'm sure that Tom will know more about this than I do. But they're pointing towards many, many, many different genes play a role, and many of the variants that play a role might be just present in the general population and quite prevalent, and maybe it's a particular combination of which ones you have together with the environment that may play a role. So it's going to be extremely tricky, I think, for those types of disorders.
Here's the next great question. These are really interesting questions. How does Darwin's theory of survival of the fittest permit so many cell mutations that cause cancer to be passed along genetically? This is really good. Looking at the panel, looking at each other.
Well, I think one thing that we should point out is, the incidence of cancer is definitely on the rise. Well, why is that? It's because we're all living longer. It's true that families can be predisposed to certain kinds of cancers because of these genes being transmitted from parent to offspring. But most cancers are the accrual of mutations that occur over time. And if you look at the incidence of many, many cancers, they just continue to go up the longer we live. And so one of the important things to bear in mind is that we tend to, although this is changing a little bit, we tend to have children well before we get these cancers. And so these kinds of changes that, really can lead to heritable predispositions are very likely to be carried along. But it's really true that the incidence of most of the common kinds of cancers really occur, they go up quite precipitously as our population ages, and so this is well beyond childbearing years, and therefore not as likely to affect selection against people that get these kinds of cancers. There's one explanation, but maybe other people would like to. And the other thing we have to bear in mind too is it's a combination of things. We are living longer, but many things in the environment are becoming increasingly a problem. As we discuss air quality, the ozone layer, exposure to UV, those things. The problems that come from environmental factors are also going up, and I think we really are beginning to make a concerted effort to address those things. But we have to bear in mind the longevity of the population and the environmental factors being big contributors to increased incidence of so many cancers. And coming back to what was pointed out is how complex these cancers are. Breast cancer is in even 4 or 5 diseases. It's hundreds of diseases. It's genetically quite complex and therefore intrinsically much more difficult to treat than many of the cardiovascular disorders. I believe we're very close to the point where cancer is going to surpass cardiovascular disease as the nation's killer or the leading cause of death in the Western world. And again, because of the complexity of cancer, it's going to continue to be very challenging. It's not going to be one cure. It's going to be hundreds and hundreds of cures.
Dr. Curran.
So I think Celeste is saying that the cancer, from the Darwinian perspective, is the way in which people like me are done away with to make space for the younger population who need resources. Let me give you one example, not from cancer, but from sickle cell disease. Sickle cell disease is very prevalent in African Americans and in Africa. The reason is that one copy of the mutant gene, which causes this terrible phenotype when it's in 2 copies, confers resistance to malaria. So when the malaria parasite infects the blood cells take the sickle shape, and then they're wiped out, taken out from the system. That helps combat the disease. So sometimes a gene variant can have an advantage as well as a disadvantage.
What happens with ApoE? Is that something that gives you an advantage, or this is a gene that if you happen to have 2 copies of one variant of it, you have a very high risk of Alzheimer's disease, and 2 copies of another variant, you have very low risk. If you have the high risk variant, does that do anything for you earlier in life? I mean, does it help you with your cholesterol or something? It was discovered with heart disease, right?
Yes.
So what does it do for you? I don't even know. Just thinking about it as you're talking.
It depends on which alleele you're talking about, but one of
The
them
bad
actually
one.
detects from heart disease.
So you can be protected from heart disease but get Alzheimer's? That's interesting. That's really interesting. So it shows a different natures. Go, Dr. FitzGerald, sorry.
Yeah, I was just going to sort of expand a little bit on what Celeste had to say, and only differ with the very last bit of what she had to say about cancer and heart disease, because it highlights again the fact that what it's all about is the interaction between the environment and our genome. That these companies that sell your genome to you, it's like one hand clapping. Okay? It's about the environment and your genome. So let's think about heart disease. Most people in the United States have heart disease, even though more money is given to cancer research. But we're not worried about that. But what's interesting is that in the wake of the Vioxx experience, the FDA started asking drug companies to perform large trials to look for these small average effects of risk before they would approve drugs for heart disease. So the reaction of drug companies, one after the other, was to move out of heart disease. Well, that's a problem in itself. So where do they go? Well, they have an emphasis on cancer and neuroscience for reasons that we understand. But because of the change in our environment, the incidence of diabetes has exploded, not just in this country, but throughout the developed world and increasingly throughout the developing world. So they switched their attention in very large measure to diabetes from heart disease. Well, the question is, what do diabetics die of? They die of heart disease. So in fact, we've taken our eye off the total problem to focus on one part of the problem. But it comes back to this issue of really never getting away from the fact that it is the integration of the environmental influence with your genomic substrate. And in heart disease, just like in those cluster of cancers and those clusters of psychiatric diseases, it's exactly the same story. So when these big studies were done to find new genes associated with heart disease, yes, we found a bunch of them now, but they all account for tiny contributions to the risk of heart disease. So it's likely that in everything you call a heart attack or a stroke, just like what you call breast cancer, there are multiple genes which may have variants that increase your risk. But the trick is, how does that interact with the environment that you're exposing yourself to, your diet, your exercise, your smoking, etc. And that's why it's so difficult to just sell single genetic test and say that this can actually predict that you're likely to have a heart attack, more or less likely, even if it's something like ApoE forms, where there's a reasonable amount of evidence as to their importance in their own right.
Here's a ethics question. Have institutional review boards and the need to, in quotes, protect human subjects limited genomic research? Have they limited, has the need to try to protect human subjects in studies limited genomic research?
Well, I think that over the years what has happened is gradual development of new rules. So initially there were questions as to whether genomic research was high risk, because risk is typically defined and thought of in terms of physical injury. When you undergo a protocol where you have you're given a medicine or a procedure is done that could result in some sort of physical risk. And something like genomic research typically is just a swab, saliva, blood, and the idea if you actually look only at the physical part of the participation in a research protocol, that the risk is negligible. And what people have suggested over time is that the other risk, of course, is informational. And personally, information might get out, or it could be that the result of a study ends up stigmatizing the population that was in the research. So there's an evolving sense of the ways in which genomic research might create problems for the participants. And I know in England they have slightly different regulations than they do in the United States, stricter in some ways, but maybe the scientists who are actually conducting the research are in a better position to say whether it actually has limited their work. My sense is that that people have been able to conduct the work they needed to, even if that required them to revise the way they had planned to do it.
I think all of us at Penn in particular find regulatory compliance as an increasing portion of our effort. We are being held to higher and higher standards to do all kinds of research, whether it's with radioactive material or toxic compounds or animal studies or getting access to patient samples. And it is a challenge, and the documentation is going up, and I think we all gripe about it. On the other hand, if the net effect is to protect patients to the best of our abilities from harm from things being done too quickly or without appropriate controls and brakes being put on things, then I think it's a price we're all pretty willing to pay. But I know we still gripe about it a lot.
This genome chip has 3 people's genotype on it. It's actually a real piece of research data. It's identified with a barcode, so you just scan it, and that scan delivers a number. And that number is in an unbreakable code. There's no way for me to link that back to the individual or the 3 individuals whose data are on this chip. So what the IRBs kind of forced us to do was to develop very good encoding and encryption systems to handle the masses of data. And we went through an evolution. I think there was a period of time when it was a somewhat difficult relationship. Now, to a large extent, IRBs partner with scientists in order to come up with the best solutions that will meet the needs of the patient population as well as the research study. And I think that part of things is working fairly well now, so I don't think there's any impediment to progress.
There's an interesting project at Harvard right now, the Personal Genome Project, where they've taken an opposite approach to a lot of the issues with working with genetic samples. And their idea is that it should all be public. All the information should be public. And the logic behind it is that, in a way, after all, any genetic information could ultimately be identified because it is your information. So even if the code is broken, logically they could figure out that it was you because it is you. If you equate yourself with your genome, it's not going to change. So given that that's a fundamental fact and that there are increasingly large biorepositories with more and more and more genetic data being stored, the sense was that it was becoming increasingly impossible to somehow guarantee that this information would be protected. And a lot of the studies being done for personalized medicine are studies that require thousands of samples. And so rather than continually evolving new and more complicated protections for the data, they turned on a dime and said, no, it's just going to be public. And part of the argument is that it will change people's sense of the importance of genetic data, the sort of over-importance that people sometimes attribute to genetic data. And although I have to say that originally they planned to have, I think it was 1,000 people by the end of 2008. They only have 10. I mean, they started intentionally with 10 people who would post their genomes online. And then they were going to enroll another group by the end of 2008, which they haven't done. And I don't know if it's just technically they haven't gotten around to that. Although it is interesting also that they can't get funding for this part of their work from the federal government. This is from a private foundation.
I think this is maybe our last question. We have such good questions. I was trying to get them all in, but we actually have more questions than we have time. Here's the last one. What are the cost-benefit ratios? Dr. Curran mentioned one chip costs $1,000. You need multiple chips per test. Who's going to cover the cost for these tests?
So the chips that I discussed are the research chips that are not being used for clinical diagnostic studies. The technology is moving ahead, and clinical studies can be defined in a different way to reduce the amount of information, because in most things you don't need the amount of information that's on the chips that I showed you. And those costs can come down quite dramatically. The cost of sequencing a genome used to be, what, over $1 billion? And now it's, I've heard quotes, it's coming close to the $1,000 level. I don't think it's quite there yet. So the cost will come down, but a clinical test is very different from a research test. And it requires a lot of validation, quality control, and verification before it be released to the public.
Yeah, I think it comes back to that issue of getting the evidence for incremental value. Because unless there's incremental value beyond what we do at the moment, third-party payers are not going to reimburse for these tests. And getting that sort of evidence is one step beyond FDA regulatory approval. And that's why this is a field where there are a lot of challenges.
Thank you all. It was a great audience, wonderful questions. And I think Dr. Eberwein has some concluding remarks.
This is the first of what we hope will be several PGFI Franklin Institute collaborations that will bring important developments in genomics to this impressive public forum. On behalf of Junhyong and myself, we would like to take a moment to thank Dr. Kristen Thiel for her considerable effort in making this symposium a reality. We also would like to thank Curran, Elinch, and Dr. Frederick Burley at the Franklin Institute for their help, guidance, and support in organizing this event. Of course, we are grateful to our colleagues in Penn who spoke so eloquently and passionately about this important topic that affects each and every one of us. We especially thank Gina Kolata for so ably moderating this event. We also want to again highlight the co-sponsorship of this event by IDMAT and the financial support of the Penn Center for Bioethics. For those of you who would like more information about this topic, there are various websites highlighted in the program that will direct you to more comprehensive resources. Finally, given the discussion of individualized medicine over the last 2 hours, I'm reminded of a children's poem that I've read to my children as they are being tucked into bed. I'm sure this is the first time Dr. Seuss has been read in this august room. But the title of this poem is Too Many Days by Dr. Seuss. Did I ever tell you that Mrs. McCabe had 23 sons and she named them all Dave? Well, she did, and that wasn't a smart thing to do. You see, when she wants one and calls out you who, come into the house, Dave, she doesn't get one. All 23 Daves of hers come on the run. This makes things quite difficult at the McCabe's, as you might imagine, with so many Daves. And often she wishes that they were born. When they were born, she had named one of them Bakken Von Horn, and one of them Goose Foos, and one of them Snim, and one of them Hotshot, and one Sonny Jim, and one of them Shadrach, and one of them Blinky, and one of them Stuffy, and one of them Stinky. Another one Putt Putt, another one Moonface, another one Marvin O'Gabble Balloonface. and one of them sneaky, and one weepy weed, and one Paris garters, and one Harris tweed. And one of them Zanzibar buck with fate, but she didn't do it, and now it's too late. With that, we thank you for joining us, and we wish you a good evening.